Yeah. That's better. Okay. So hi everyone, as Jimmy mentioned, my name is Cody Chacon, but I publish under my maiden name, Cody Ching. So I live kind of like a double life. And today I'll be talking about understanding Amphidromy and fish community on Oahu, Hawaii. So just a little bit of an introduction. As Jamie mentioned, I started the animal science program in 2016. And initially I was a pre-vet student, but that all changed in 2019 wherein I did a study abroad in South Africa. And being with all these wild animals in their natural habitat really made me want to pursue conservation instead. So after that, I volunteered with a USDA field biologist, and that really solidified my decision to want to go towards research for conservation. Then in 2020, I graduated with my Bachelor's of Science in Animal Sciences, and then I started my Master's NREM Plan A program that fall. And then 2022, I graduated. And I was very fortunate to get a state job just a few months after graduating as a hydrologist with the Commission on Water Resource Management. And I'll kind of go into that a little bit at the end of my presentation. So just to start off, sorry. So just to start off, we only have a few native Hawaiian stream species. If you're not familiar with our native streams, we have 5 Oahu, that's just 5 native gobies, and then we have 2 mollusks, and then 2 crustaceans. So we have a very small native population in our streams. So for the first half of my presentation, I'll be talking about my master's thesis, where I was uncovering migratory patterns of Oahu nakea, so one of our native stream gobies. Now, traditionally, it was thought that they exhibit an obligate amphibious life cycle. This means that the adults will live and breed and lay eggs in freshwater streams. The eggs will then hatch, and then the larvae will flow downstream into the ocean. Well, they'll live as pelagic plankton for several months before migrating back up the streams. And for them to migrate up the streams, they use a fused pelvic fin. It kind of looks like a suction cup on their chest, and they can actually climb waterfalls with that. Recently, there have been a few papers that came out that are saying Oahu nakea actually exhibits facultative amphidromy. This means that some individuals of the species do not need to go to the ocean and they can complete their entire life cycle in fresh water. So Hogan et al. 2014 found that 62% of their Oahu nakea population completed the entire life cycle in fresh water. And then more recently, Lisi et al. 2022 found 83% completed their entire life cycle in fresh water. Now, how do you know if an individual was in freshwater or saltwater as a larvae? Well, you have to kind of look at their otoliths. And these are super cool. They're calcium carbonate structures. And specifically, we'll be looking at the sagittal otolith, which is the largest otolith. They kind of grow like rings on a tree. So each ring will represent one day of life. So the center rings are going to represent when that fish was a larvae, potentially in salt water. And then the outer rings represent when this fish was an adult or in freshwater. And you can tell the 2 different regions apart by the metamorphic check, which is that thicker dark band as indicated by the white arrow. And this is just deposited when that fish is undergoing that metamorphosis, where it's changing from a pelagic larvae to now a benthic goby. So now we have the 2 different regions distinguished. We are going to be looking at strontium, and this is because it's significantly higher in saltwater compared to freshwater. So when we analyze the center region of the otolith for the strontium concentration, if we see a peak, that means that this fish was in saltwater as a larvae. So for my master's thesis, these are my study sites. I chose 3 different streams on the windward side of Oahu. Kahana, Kahalu'u, and Waimanalo. And we tried to choose watersheds along a rainfall gradient to look at different environmental factors that may cause a species or an individual to want to migrate versus to stay in fresh water its entire life cycle. So I collected 30 fish per stream with a sample size of 90 total. For the environmental factors we looked at, we had several. I also collected water samples in the streams to verify the strontium concentration in that particular stream. And looking at the MNDS plot, you can see that the different streams did have different types of environmental factors between them. So after my fish were caught, I had to extract the otoliths. And each fish will have 2 otoliths. And I was able to successfully extract 178 otoliths from my 90 individuals. I unfortunately lost 2, which really bugged me because I wanted that 100%. But at least I got 1 per 90 individual. And we're going to be looking at 2 different ways of preparing the otoliths now. So the first method is going to be the transect method. And this is the more traditional method that's used and the same method that the previous papers used as well. We're going to be polishing the otoliths to kind of get to that center region because we want to analyze the area of the otolith where the fish was a larvae. So if we look at the diagram, that's going to be that blue shaded section. And you can kind of think of it like layers on an onion. You kind of have to like cut it away to get to the center. And then the laser kind of just goes right across the surface, as you can see in the bottom photo. And then the other method we're going to be trying out is the depth profile method. And this is the method where the otoliths are just kept whole and you don't do any polishing and the laser kind of just drills through the center. And the reason why you can do this method for this species of otolith is because these otoliths are so translucent, you can still see that metamorphic check even without polishing. So we are sure that we are analyzing the correct section of the otolith. We use this method to kind of just verify the transect method. So again, each fish has 2 otoliths. One, we use the transect method, and then we just verified that results using the depth profile method on the other otolith for the same fish. So for some results, with the transect method, we first started off with a small subsample of 40 otoliths from 40 different individuals. Of that 40, 17 showed evidence of marine migration, and this is indicated by a strontium calcium concentration greater than or equal to 4 millimoles per mole. As you can see on the graph, it's quite clear whether or not that fish was in saltwater versus freshwater because there is a very distinct difference in the strontium concentration. So 17 showed evidence of marine migration, which means 23 showed no evidence. And does that mean that they potentially stayed in fresh water their whole life? Well, we analyzed that using the depth profile method. And actually, the other otolith from those 23 showed marine evidence of marine migration. So once it was determined that the depth profile method was more sensitive in detecting that marine migration, we analyzed the remaining 50 individuals, and all of those also showed evidence of marine migration. Means we have 100% of my individuals showing evidence that they actually did migrate from the ocean. So based on the results, I think we should reconsider obligate amphidromy for this species because 100% of the otoliths showed evidence of a marine larval stage, regardless of the distinct environmental factors between the study sites. And this is important for the life cycle because it also facilitates colonization. So the fish can move between the different streams within an island and even between islands as well. The Hawaiian streams also have major hydrological variations. I'm sure you guys have all gotten flash flood warnings before. The streams definitely rise very quickly during high flow events, and that can physically push out the larvae. So there have been no ecological verification of larvae in our freshwater streams either. They just don't have that suction cup pelvic fin yet. That happens when they undergo that metamorphosis. So they physically cannot stay in our streams, especially because they are so flashy. It's also important to reconsider obligate amphidromy because this change of classification from obligate to facultative amphidromy could change management and conservation actions. So, for example, if people are hearing, oh, majority of Oahu are able to complete their entire life cycle in freshwater streams, it's not important to actually have the streams connect to the ocean. But here we see that it's very important, it's quite necessary that our streams connect to the ocean in order for our native species to complete their entire life cycle. So going into a little bit of why we might have these contrasting results compared to previous papers. It's the main difference in the otolith preparation methods. It's the transect method versus the depth profile method. The transect method, you're polishing the otolith, whereas the depth profile method, you just keep the otolith whole. So if we look at this graph, this is actually two otoliths from the same fish. When preparing the one otolith using the transect method, there isn't a high peak in strontium concentration, so you would first interpret this as this individual not leaving the freshwater stream at all as a larvae. However, when we analyze the other otolith of the same individual with the depth profile method, we see a clear saltwater signal that this fish actually did migrate from the ocean. And one reason why we could have these different results too is because these otoliths are so microscopic. So if we look at the bottom picture, that's one microscope slide, and each square has one tiny white dot in it. That's an otolith. So it can be quite easy to accidentally over polish it, and there goes the center section, or under polish it and you didn't expose it. So you're only analyzing the area that has just the freshwater signal. And if you are interested in my research, we got this published last year in the Journal of Fish Biology. There's more details and information there if you want to explore further. So switching gears, I'm going to now be talking about my current job as a hydrologist working for the Commission on Water Resource Management. The Commission on Water Resource Management administers the State Water Code, which was created by the 1987 Hawaii State Legislature. The Commission's mission is to protect and manage the waters of the state of Hawaii for present and future generations. I work in the Stream Protection and Management branch of the Commission. Super cute, we're called the SPAM branch. And we regulate the state's surface water resources. That just means like all the water you can see on the surface of the earth, not groundwater. And we permit stream channel alterations and diversions statewide. And this allows us to assess the potential impacts of the channel alteration or the diversion. So if you want to take water from the stream, you're going to have to apply for a permit first, because we need to see how much water you're going to take and how is that going to affect any downstream users as well as the natural ecosystem in your stream. Part of my job is to help maintain stream gauges quarterly. We have about 50 across the entire state that we have to visit 4 times a year with a field team of about 3 people. So we do a lot of field work. For example, I'll be on Maui tomorrow. Mainly just day trips. And then when we do these trips, we are also taking stream flow measurements and doing data downloads. So we have different types of stream gauges. For example, we have pressure transducers, which are in the stream, and we have to physically go there and download the data from the transducer. And then we also have real-time stations where the data is connected to a satellite, and then we can see it on the computer in real time. But all of these gauges need to be calibrated with a stream flow measurement, because you can get shifts in the stream. For example, if big boulders change the gauging pool, you're going to have a different water level, and that just all needs to be calibrated with the stream flow measurement in the field. In addition to maintaining the stream gauges, I also help with snorkel biota surveys, and we do these on East Maui, West Maui, and Oahu. And then for the rest of this presentation, I'll be going into some of the snorkel surveys we did on Oahu. So last year we did a total of 25 surveys on the windward side of Oahu in 6 different streams. We went to He'eia, Waihe'e, Waiahole, Waikane, Kahana, and Punalu'u. And we tried to get 4 different elevations within each stream. And then we also make sure to get streams with USGS stream flow gauges. So we have that historic hydrological data to go with the biota survey as well. The survey method we used is a modified point quadrat method. We're going to have transects that are 10 meters apart, and then we're going to have a total of 20 quadrats, so it's going to be a survey reach of 200 meters. At each transect, there's going to be quadrats that are randomized. You're going to either survey the left, middle, or the right stream bank. And then you basically have to like visualize a square of where you're going to count the fish in. So I put my head in the water and we stay there for 2 minutes and we just count how many fish swim in the quadrat. We estimate their size as well. And then we just keep moving upstream until we hit the total 20 quadrats. We also looked at different environmental factors. For example, canopy percent cover, which we estimated using an app. As you can see in the picture, you kind of just tell the app what is canopy and it highlights that in pink and it'll give you a percentage. We did that every other transect and then we averaged it to get a average canopy percent cover for the whole survey reach. We also looked at velocity, wetted width, and active channel width. Wetted width is just how wide the stream is currently, and then active channel width is how wide is the stream going to be during like a high flow event. We also looked at the maximum depth, elevation, substrate percentage, and then the different substrate categories we have are bedrock, boulder, cobble, gravel, sand, silt, and organic matter. And then for the habitat types, we just classified them into 3 different types, riffle, run, and pool. We also had some cascades and some side pools, but we just reclassified them as riffle and pool respectively. And then I looked at all these environmental factors and I tried to see which ones were most significant with density. And we have 3 that came up. So depth, elevation, and percent boulder. And I'll be going into those results. So for depth, this is kind of the format of the graphs I'll be using moving forward. So the y-axis is going to be our environmental factors and the species are going to be on the bottom. Exotics are on the right side and then natives are on the left. So we can see that the exotic species have a wide range of depths that they can be found in. And we can see that the depth of range overlaps with our native species. So we do have kind of similar habitats as far as depth goes between our exotic species and our native species. For elevation, again, the most exotic species have a wide range of elevations that they can be found in. However, the native species range is not as wide. We can see that certain species prefer certain elevations. And this kind of makes sense too, because for example, if we're talking about our Oahu species, there is one Oahu named Akupa, and they actually don't have that suction cup pelvic fin, so they're not as good climbers, so they hang out at the lower sections. And then we have Oahu Alamo'o, who's a really good climber, and most of the time they're seen at the higher elevations. So the native species tend to naturally just orient and distribute themselves throughout the stream reach to kind of avoid that overlapping competition. And then we can see at the lower elevations, our exotic fish species tends to overlap with our Oahu species. And then at the higher elevations, our opie overlaps with the exotic invertebrates. So our opie is our native mountain shrimp. They're really good climbers too. And we can see that they're kind of competing in the higher elevations with some other exotic invertebrates, such as the grass shrimp or like phycid snails. Moving on to boulder percentage, again, we see the same trend. Most exotic species have a wide range, and it's because these exotic species are also generalists. They can live in differing types of environmental factors. And then we see that our native species tend to have more preferences. There is less overlap between the species, and they do have more of a niche habitat. So for example, Nopili likes to farm algae, and they need the boulders because that's where the algae grows. So you tend to see like certain species in certain habitats just based on like their lifestyle or their diet. Okay, so just some take-home points from the biota surveys. Cicelids and prawns were seen at all streams. showing that these are kind of like the most common exotic species that were seen in these biota surveys. One thing to note about the prawns is that they are amphidromous, so they have that same life cycle where they migrate from the ocean, which makes them kind of harder to get rid of as they can reestablish in the similar way as our native species can. Unfortunately, on Oahu, we didn't run into any alamo'o. I have seen some on Kauai and on Maui, but on Oahu, there aren't as many, even though we looked at some higher elevation reaches. So that's kind of unfortunate. And then just noting that elevation, depth, and percent boulder had the most significance with the species densities. And we can also note that the native species have environmental niches between them, and it's important to see where those environmental niches are and how they overlap with the same habitats that the exotic species are living in, and we can kind of see how or if there's any competition between the species for resources. So thank you so much for listening to my presentation. I have my email if you want to contact me for any questions later on. Thank you.